explain to me why, in your opinion, does a study of the brain need philosophy? What does it add?
That's a great question, because I'm not quite sure exactly what philosophy adds to neuroscience. I know what neuroscience adds to philosophy, and that's really what I came into. But I do think that neuroscience can benefit from people who spend a lot of time reflecting, especially on some things that often get left assumed in neuroscience. So one of the experiences I've had throughout my career, I mean, going back to graduate school, was that when I would engage with people, I mean, it was great. They'd teach me how to do electrophysiology and optogenetics and all these cool things. But when I was engaging with them, it also seemed to be the case that they would start talking about some things that they often just kind of left implicit in practice. And they agreed, I think, that kind of making those kinds of implicit practices more explicit led them to think more about them, about things that they just kind of routinely assumed in their day-to-day laboratory practice.
Right. Give an example of that, where what might be an assumption that a neuroscientist make, where a philosopher would be, well, take a second thought of that.
Well, there's one that's going on right now. So I've got a new collaborator at the University of Tennessee Health Sciences Center, Rob Williams. Rob has had a monumental career. He basically trained as an electrophysiologist in the Hubel tradition, the Hubel and Wiesel tradition. And then he was one of the first to start doing some work in neurogenetics. And so he's just had an incredible career. And in one of the things that we started talking about when I first met him a little over a year ago, we started talking about was he's become convinced that our whole notion of the way the brain keeps track of time is completely wrong, and it's led us down so many paths, synchrony and synchronous firing and synchronous activity, that it just turned out to not really deliver on their initial promises that he and I have started kind of reflecting on, and he's come up with a new notion, which I'm not quite sure he's fleshed out yet, but he calls it parasynchrony. And it's just one of those things that was just kind of sitting there in the background in some of the things he had been working on, and all of a sudden now it's in the foreground, and he's thinking harder about it, and he's going to come down to my annual conference this year and give us a talk on how far he's getting. So
cool. So what is that? So what is the evidence that he's come across recently that makes him think that we've got this time tracking mechanism incorrect in the brain?
I think that what he's been looking at, so this is really his gig, but I've been talking with him about a little bit, is just the fact that you have the brain operating on so many different time scales. I mean, everything from literal less than milliseconds all the way up to, pronounced activity that take place over a number of seconds. And so how in the world, so what he seems to be driving at is that there just isn't this kind of clock time being keeping track of in the brain. The brain keeps track of a lot of different time scales, and they're often they interact with these time scales, interact with each other. These brain activities interact with each other, but the idea that there's this kind of wall clock time in the brain, which a lot of the work on synchrony and synchronous firing just assumes, probably is mistaken.
Interesting. So that would also play into things like circadian rhythm biology.
Yes.
And I guess that's really the point that you're making at, is that neuroscience now has layered assumptions and hierarchical theories that we have based on each other, and they're implicit in each other. But what something philosophy is quite good at is really poking holes at the underlying structure. That be correct?
Well, I'd say bringing it out in the open, asking the kinds of questions that actually bring some of these assumptions or some of these implicit practices into the foreground. And I think that's one of the things that philosophy can do. So I recently did some work on this with so the European Journal of Neuroscience is doing a special issue on what can a philosopher's toolkit contribute to progress in neuroscience. And so, my kind of meta-scientific approach to a question like that is, let's ask some neuroscientists and see what they think. So I gathered together a lot of people that I have collaborated with over the years. Alcino Silva. I got Rob involved since he's my new collaboration. Mike Lehman, Pat Churchland. I got Pat involved in this. And then some younger people that I've been interacting with. My colleague at University of Mississippi Medical Center, Brad Walters, a David Parker, a physiologist at Cambridge. And I just put the question to them. What do you think a philosopher's toolkit can contribute to neuroscientific progress? And the result was just these hours of transcripts of interviews. And what I tried to do then was pull out of this some of the ideas. And I think it's a really encouraging paper, because anyone who is a philosopher who has interest in interacting with neuroscientists, this gives you some evidence that the neuroscientists, at least this group of them, will take you seriously, and that they do think that there are some problems that people that think in a more abstract, more reflective way, that are in some sense a step away from the day-to-day turmoil and hullabaloo in the lab, can help by reflecting on.
Right. That makes a lot of sense to me. I did a presentation in my neuroscience degree about consciousness. And at times, that's probably one of the best examples of where you're reading something and you're really not sure what field it is classified into. It doesn't fit into a neat bucket. But a lot of times it didn't felt like what I would say is classical neuroscience. Like molecules, electrons, these physical processes, mechanisms, right?
Yes.
A lot of the time it seemed a lot more philosophical.
Yeah. And generally what that means is, it means that you're opening the arena to other concerns, other considerations, always with rationality and rational argument first and foremost. But nevertheless, you're giving play to some ideas and some reasoning that it's just simply not time to do when you're engaged when you're running an experiment or when you're crunching data from experiments.
Right. Do you think it's always in areas where we are ignorant? Because, we're massively ignorant to consciousness in terms of what's actually happening in the brain. So do you think that that's the kind of the place for philosophy, is when it just doesn't really fit into these neat biological buckets? We just don't know how to characterize it. It doesn't fit into, we don't have any causal mechanism, anything on a real molecular level. That's really where the conversation of philosophy starts. That's
one place, and that's clearly one place where philosophers, I think, can contribute. But another place I think that philosophers can contribute is something that's a much, much more common ground for neuroscientists, and that is just kind of reflecting upon science and practice. What exactly are you doing when you are designing your next experiment? What exactly are you, what goes into the process of experiment planning? That was the purpose of Alcino's and my and Tony Landris' 2014 book, where we took the first 20 years of molecular and cellular cognition, a remarkably productive science, and basically said, okay, let's call from this the landmark papers, and we used a really simple measure. Basically, we were just looking at, citation measures, just to give us get an idea, not necessarily of the best science, because citation measures can't measure that, but of the impactful science, the science that people were using and hence referring to in their publications. And so, and then what we did was we said, okay, now let's look at this group of papers, and let's try to find what's common, not among the ideas, but among the kinds of experimental designs. And so from that, we kind of culled out what we called a framework for scientific experiments. And then we looked specifically, since we were looking at molecular and cellular cognition, we looked at what we called connection experiments, which are just experiments in which you're trying to establish a causal relationship between two neurobiological phenomena. phenomena. And then we started saying, okay, what do we find common in these kinds of experiments? And that led us to what we discussed in that book, the kind of ways in which different experiments are designed to achieve different purposes for establishing a causal relation, and then more importantly, the way they're integrated across experiments. And so that was the whole purpose of that book. But we also articulated what we called, the tool development experiments. And once we finished the book on connection experiments, that's the next thing I launched into. What's going on in these kinds of experiments where neuroscientists are trying to develop and validate new research tools? And that's been just a gold mine of ideas for the last decade.
You want to share some of those critical insights? Like when you dove into the different methodologies that are used in neuroscience, did you upturn anything that you thought was, a big flaw, a big issue, something that was critically wrong with the field, or something that needs maybe rethinking or reconstruction?
Yeah, well, so one of the things that we were trying to do in the book was really trying to be as descriptive as possible. Really just trying to get a grip on how these experiments are designed and then how they're executed. And then once we feel like once we got that, then we think that then you're going to be better set to do the kind of normative work that you're talking about. But until you know exactly how these practices work and what they've delivered, it seems to me that reflecting normatively on science isn't going to be very helpful because you're just not going to know, you're not going to have in place a kind of good picture of how the field actually works. So now when it comes to like these connection experiments, one of the things that became absolutely critical that we saw early on was that experiments really are not designed one at a time. I mean, they're designed in terms of research programs, at least among the papers that turn out to be the most impactful on the field. They're a piece of a broader picture. Some of the pieces are already in place, but there's pieces missing. And so these experiments are actually designed to be, if successful, the results integratable directly into the ongoing kind of causal pathway that gets us from wherever we're doing our manipulation out to the behavior we're measuring. And I think that's a really important thing to notice, because I think at least so much of the philosophy of science is focused on just single experiments. And I think a lot of the methodology in science focuses on that, missing the idea that a lot of these experiments are explicitly being designed so their results, if successful, will plot right into these ongoing research programs. And I think that's a really important insight to have, because it gives you a way of answering questions like, well, why in the world in molecular and cellular cognition do most people remain content with some relatively simple measures of behavior? Why is contextual conditioning, just constantly used over and over again as an experimental protocol? Why is fear conditioning so constantly used? Well, the reason is not because it's easy to do but because once you find those results, they will fit right in with all the other results that had been gathered using that particular protocol. And
right.
That, I think, is a crucially important thing to understand if you're going to understand neuroscientific practice.
Right.
And probably broader scientific practice too. I mean, I won't speak for physics or sociology, but I think you'd find exactly the same thing if you looked hard metascientifically at like molecular biology, for example.
Yeah, it makes complete sense. What do you think are some of the biggest pitfalls or traps that we fall into with a system like that, where we're kind of just building on each other's results, and maybe there's a lot of implicit bias that are building into those results?
Yeah.
Like, talk around that a little bit. What are those big pitfalls?
Well, I think we're only going to find those in what's being left implicit if we're carefully reflecting on what's actually being done.
Right.
Now, of course, look. One of the things you always going to have to do is you're going to have to worry about the broader implications of what you're doing. You're not really studying learning and memory. I mean, you're studying learning and memory operationalized in the following way to do rodent behavior. I mean, and now look, I think we can also take comfort in the idea that when it comes to those particular molecules that molecular and cellular cognition has revealed to be so crucial for the operationalizations of learning and memory that have taken place with the rodents. I mean, they're with a few little changes, for all intents and purposes, at least in the functional units, a mouse protein kinase A is pretty much a human protein kinase A. I mean, calmodulin kinase 2. I mean, so when you're actually dealing with manipulation of molecules of that particular sort of range, there's a lot of commonality especially across mammals, but even going back to sea slugs and fruit flies. I mean, so that's that. But nevertheless, you have to be really careful. What you have shown the mechanisms of a particular kind of learning and memory that you've operationalized in the following ways. And saying, now it's easy to kind of slip into saying, oh yeah, we found the mechanisms of learning and memory, but you've only found it for the particular kinds of learning and memory that you've operationalized in these.
Right.
If you enjoyed that short podcast clip, then I'm so confident you will enjoy even more the full conversation. You can click it here. Click it. Come on. You know you want to. Click it.